Welcome to Csec Biology TCP. I am Mr. Wilson from TCP Academy, and today we're looking at the May-June 2016 biology paper. It is of course the paper 2 paper, which is slated to last 2.5 hours if you were supposed to do it, but we're going to see how quickly we can get it done. Please ensure that when you go to the exam, you read all the instructions on the paper so that you'll be answering what is asked and not what you think was asked or should have been asked. Now, question number 1 is usually a lab question, so it's very important that your lab skills are up to par. Let us read this question. It comes from section A of your paper. It says, answer all questions in this section. Write your answers in the space provided in this booklet. 1A. So this is section A of question number 1. It says students investigate the effect of temperature on the rate of reaction of the enzyme catalase. Now here you'll realize that in the first sentence you can find the aim for this experiment. They use crushed potato as a source of enzyme and place 5 milligrams into each of 7 test tubes. Here you'll realize that of course potato has protein if it contains enzymes. They added 3 centimeter cube of 1% hydrogen peroxide, that is H2O2, to each test tube and place them into water bath kept at the following temperature, and the temperature are as outlined: 0 degrees Celsius, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40, 60 degrees Celsius, and of course 50 degrees Celsius. They count the number of bubbles released per minute from each test tube and recorded the data in a table. Now this is a very interesting table. Let us look at what a table entails. Here you'll observe it has a title at the top, and it has header column. So here we have the header column, header column, header column, and this would be considered the header row. The first thing here, header row, that type of a thing. And this is pretty much a convention for a table. If you observe, the table has 4 sides and the numbers are all written in straight line coming down, and there's no missing cell in the table or no missing data in the table. If you observe, the unit is not repeated in the cell here, so we have degrees Celsius and it's not repeated here. And we have oxygen bubbles, and we really don't repeat the units in the cell, we just place the numbers there. Now we're looking at the table, and the convention for table is that the independent variable, that's a variable that the experimenter will manipulate, is going to be in the leftmost column. Now if we were supposed to use the leftmost column here, which is test tube, we would have to have a legend for the graph. That would be a little more work. So I want to go on down to look at what we would have done. Here I would have just drawn a sketch of what the graph is supposed to be based on what I'm seeing here with the table. You're seeing that we're going up 0 up to 30, 40, 50, 60, but not that there. We're looking at what happens on the frequency. So we go here and we observe that we went up to about 15 there, so up about to 15, and then we had a crash. So let us look at what the question is saying. Great. So we're supposed to plot a graph on the grid provided on page 5 to show the relationship between the number of bubbles and of course temperature. So here we're seeing 2 things. It tells us what we're supposed to plot. We're supposed to plot number of bubbles versus temperature. Here it is very important that we understand the difference between the independent and the dependent variable, because it clearly says bubbles and temperature. So some students would put bubbles on the y-axis or on the x-axis and not really sure what they should do. But please be reminded, usually it is the leftmost that goes on the x-axis. So here it would have been temperature. Now if you observe here, we are manipulating temperature because we have different temperature, and we, the experimenter, actually decided what the temperature would have been. On the y-axis, this is not up to the experimenter. This will change as a result of the experimenter changing what is on the x-axis. So the bubbles are going to be on your y-axis, and of course temperature on the x-axis. That's the first thing that we need to know. And for that, you would have gotten 4 marks. But here we're supposed to get this information from the graph. We're supposed to determine the optimum temperature for the enzyme. Now, the optimum temperature is, of course, going to be the ice point at which the enzyme works and is efficient. That would have been right here at the top. And if we want to find that, all we have to do is to look for the highest number of bubbles, and that would have been 15. So it's 15, and the temperature here would have been 40. This is, of course, the optimum, the ice efficient point, and that's right there at the top of that graph I've just drawn to explain before we opt. So 40 there we have as the answer, 40 degrees Celsius to be specific. The unit is very important. Now, working with graph is a lesson by itself, and students tend to shy away from graph because I don't know if enough work is not done there, but they're still shying away from graph. So the first thing we had to do, we had to work out the axis for this graph. But we did some things that probably would have been easier to be done before the axis. One of the things that we did, we drew the graph. So if you observe this red line coming down, I'll just show that to you. This red line coming down, showing that we drew the graph, and you're seeing the directional line, meaning that it is increasing in this way. So we drew the graph rather than just populating the grid. You're seeing the red line going all around, showing the x and of course y axis, and then you'll see the axis being a name. Temperature here in degrees Celsius, it is measured, and on this side you're seeing that it is measured in the number of bubbles per minute. And then we had to represent this on the graph paper. So what we had to do, we had to work a scale out. So here we're saying each of these grid represent a centimeter. So 1 grid, 2 grid. So we're saying here 2 centimeter represents 10 degrees Celsius, and you're going to see that in the scale. Here we're saying that 2 centimeter represents, this probably is represented differently than it is here. We're saying that 1 centimeter represents 5 degrees Celsius, and on the x-axis and on the y-axis 1 centimeter would of course represent 1 bubble, and that you can pick that information up on the table. Now we get into drawing the curve. Now all lines on a graph, they are referred to as curve. Now if you're using a dot to plot the points, then you want to ensure that the dot, they are all circled. So I'm using dots here, so I'm circling just to identify the point that I am supposed to swing the curve through. So you'll observe here that the dot is all there. I'm using Microsoft Word to draw this graph, so it might not be as if you were drawing with your pencil. So the line is all swung pretty much through all these points, and it comes up at the top here. If you observe, there's not a sharp decline, there's a little curve here coming down, and that's pretty much, you should always have this little curve. It didn't just happen that boop, went up like a ball and returned down. It had to sort of slow down and then came right back down to the bottom. So this pretty much represents what your graph should be, and I'm just going to just let that zoom in a little bit so you can have a better appreciation of what the entire graph should look like. Just let me show you the full graph here before we move on. So I'm going to put everything on one page to show you what a graph should look like if you are doing the graph. So that's pretty much what your graph should look like for your exam. You want to ensure. Now, another thing that a graph should have is a title, and you observe the title is given here, so there's no need to go recreating the title because it's already given. So all graph must have a title, and the title is given here, so there's no need to go recreating the title. I do hope that you have that and you are able to do your graph question should you see one for your exam. So we move on to the other questions, and students tend to say, I would want to see the question and not the answer. So we're going to try as much as possible to see how best we can do that. So explain the shape of a graph at each of a following temperature range. So we want to explain the shape of the graph. In explaining the shape of the graph, two things might be captured. Why is the graph shaped like it is shaped? And what does the shape actually means? So we want to look at, in explaining the shape of a graph, we want to know why is it shaped like that and what this shape means. These two things we need to take in consideration. So let us look at what the answer is for this particular question. So we're looking at that, 0 to 10 degrees Celsius. There was a gentle increase in enzyme activity, which is pretty much a common thing when we are looking at curves like the sigmoid curve. It starts out very, very slow and then picks up. So there was a gentle increase in enzyme activity, resulting in a gentle increase in curve of the graph, or in the curve of a graph, which showed, of course, an upward movement. So we're looking at what happened there, which showed an upward movement from 0 to 1 bubble. So there wasn't any bubble, then we had 1 bubble at about 0 to 10 degrees Celsius. That's how we explain the shape of a graph. We're looking at what is happening and what would have caused that to happen. Let's move on down to the other question. Between 11 and 40, so remember 11 is just 1 block away from 10. Remember the graph we were looking at. There was a sharp increase in enzyme activity. And let's up on right back to the graph to look at that sharp increase. So here we're looking at 0 to 10. So let's just starting off. But then when we look there, we're seeing something more like an exponential growth. The sharp increase, diagonal line going up. So we said there, for 11 to 10, there was a sharp increase in enzyme activity. And of course, that resulted in a movement up to 40 degrees Celsius, which was, of course, the optimum temperature. Now, this is reflected in the sharp increase in the number of bubbles from little over 1 bubble to 15, and the sharp upward movement of the curve. So you're seeing that the enzyme there had increased, a sharp increase in activity, and wow, we moved from 1 right up to 15. Now, what is it saying here for 40? So this is when it got to the optimum point at the top there. If I can just draw that for you very quickly. When it gets to the top there, what would have happened? So this is going to be the graph here. So when it got to the top, we want to find out what happened there. So look at what happened. It went up pretty much like that, and then it came down pretty much like that. So we want to know what happened at 40, and that is going to be somewhere there. All right, great. So above 40, and this would have been 40 right at the base here. So above 40, what happened? We want to know what happened. So here we were increasing, but now we are decreasing pretty much at the same rate that we increase. So enzyme activity, having reached optimum at the high point of the graph here, took a sharp decline, sharp decline here, as temperature was no longer favorable for the reaction. We'll understand that enzymes tend to work at a narrow temperature, and if it goes above or below that temperature range that the enzyme works, what is going to happen is that the enzyme is going to be denatured. Please be reminded that enzymes are not organisms, so they cannot be killed. Now this created a non-functional active site to the substrate. That's a substrate to enzyme. Some connection wasn't taking place there. The reaction just could not continue. This is observed in the sharp decline in the curve to 0. So there was a real decline in enzyme activity as a result of the enzymes not being matching favorably with the active site, as the temperature would have denatured these enzymes. Another thing we needed to look at, we were supposed to write a conclusion that the students could draw from the result. Now this is going to be tricky because you want to pay attention as to what is asked by the experiment. So we are going to roll right back up to the aim of the experiment. We need to know that in order to write a conclusion. So the aim of the experiment here is to determine the effect of temperature on the rate of reaction of the enzyme. So we are looking at the rate, how the varying temperature affected the rate of enzyme activity. We are not looking at the temperature on enzymes. That's not what we're looking at. We are looking at the temperature on the rate of reaction. Hence, the conclusion should be fitting to the rate of reaction and not just to enzyme activity. So what did we write for this? We had something a little long. One of the things we observed from the experiment is that the enzyme work best at a particular temperature. We also observed that as the temperature increased, enzyme activity increased to a point of optimum, after which the enzyme activity stopped with the added increase in temperature. So the conclusion that can be drawn is that enzyme work best pretty much at a particular temperature, and if we increase temperature on enzyme as it were here, the activity will increase, but it will get to a point that it will eventually stop as the enzymes are denatured. So pay attention to your conclusion there. We're moving on to the other question, and I did promise that I'm just going to try to show you the question only. So just one reason why living organisms need to maintain a constant internal temperature. Well, all organisms, or most organisms, would have a constant internal temperature for the most part. And we're looking at homothermic organisms. Here we're not looking at the poikilotherms, which regulate their temperature using external means. Homotherms, they regulate their temperature from within. Now, a lot could be said here. To argue this point, a whole lot could be said. But this is what we said. To ensure that the body functions properly, as enzymes work best at a narrow temperature range. And if the temperature is off, it will affect the functioning of the enzyme. So you understand that the body is pretty much driven by enzyme activity, as reactions are always taking place. Now, if these reactions fail to take place, then you will become sick. You are not able to do what you are supposed to do. And you as a person will become unreliable, because you're supposed to turn up this morning. You can't turn up because the enzyme activities are not functioning. What the enzymes should do, it's not happening. The body should do this, the body should do that. It's just not happening. You have this temperature up and down, and it is affecting the enzyme reaction as is seen on the graph. You want to keep it pretty much at that range where things are constant, and of course you can be reliable to yourself and of course to others. What is the other question asking for? So it wants us to look at, we want to describe 2 precautions that the student should take when doing this experiment. Now, precaution seems to be a troubled word in science. Precaution here is looking at things that you can do or should do, or things, it looks at things that you can do or should be done to ensure that the experiment is done properly. In some cases, it is talking about things that are limiting factor to the experiment, that those that you can't control. So there's this controversy. I'm not too sure how you will explain it, but this is how we did. There's still this unresolved point among many teachers as to what precaution really is. Now, we want to maintain that specific temperature to ensure that there is, of course, accurate results. So that's a precaution. You want to ensure that what you're doing is right so that the result can be right. And we want to, of course, observe the experiment carefully to ensure an accurate count of the bubbles, or else we're not going to be having a good curve. So describe 2 precautions the student should take. So here it is something that the student should do to ensure that the experiment go as planned. So here they need to maintain that temperature, and of course they need to do that count properly. This was easy, I think. How could this experiment be modified to investigate the effect of pH on enzyme activity? Now you are modifying this experiment pretty much to see how varying pH would affect the rate of enzyme activity. So the first thing that we need to do, we need to use varying pH in each of the 7 test tubes. Rather than using different temperature, we're going to be using different pH. Now another thing we want to do is to maintain the temperature somewhere around room temperature for all 7 test tubes. All but temperature from the previous experiment is maintained. So we're going to keep the crushed Irish potato. We're going to keep the 1% hydrogen peroxide solution. We're going to put all this in the mixture. We're just going to change temperature, and in this case we're going to be putting in pH and varying pH. We're going to use a pH meter or pH paper to test the pH of each tube, and of course we want to ensure that we count the number of bubbles for each test tube. Pretty much the same, like I said, just to change out that which is of course temperature into pH and to maintain temperature for the same for all these test tubes, and you would have manipulated the experiment for pH. Now we're supposed to name 2 enzymes that function in protein digestion and of course describe the role of each. Now for CXC, 3 enzymes are going to come to mind very quickly. The first is going to be pepsin. Everybody will say pepsin. The second is going to be renin. Everybody will say renin. And the third is going to be trypsin. Most persons won't remember trypsin, as you're going to find trypsin being secreted in the pancreatic juice and of course found in the small intestine. So pepsin is found in the stomach. It's pretty much an acidic environment, and it breaks down protein into peptones. Not protein to amino acid, but instead protein into peptones. Then you are going to have trypsin that is secreted by the pancreas, and it works best in a basic environment, and it converts those peptones that were broken down by the pepsin into polypeptides. So there we would have answered the question. But there is also renin, and you can tell me in the chat, what is it that renin do as an enzyme acting on protein? Leave that in the comment section. That's for you to show that you have been following the paper here and you are pretty much a part of what we do. So we're looking at the other question here, C of question number 1. Before food can be chemically digested by enzyme, it must first be mechanically broken down in the alimentary canal. Now here you're supposed to name 2 parts of the alimentary canal that are responsible for the mechanical digestion of the food. Now 2 parts, most persons will pop up very quickly. One, they're going to talk about the mouth and the teeth, and they're going to talk about the stomach as the stomach is doing churning. But also as the food moves through the gut and peristalsis takes place, that is also going to help with some mechanical breaking down of a food and of course pushing the food backward. So here we look at the mouth and the teeth in breaking down the food, and the stomach. Those are very important, but of course peristalsis would also serve a major role here. Now give 2 reasons why mechanical digestion of food is important. Now of course it increases the surface area for food particles to be chemically digested or for chemical digestion. Another thing is that it helps to move food through the alimentary canal in a small manageable portion. You really just can't take a dumpling if you were in Jamaica and just swallow that dumpling. You need to get it into smaller parts that it can move through the alimentary canal with great ease. The other thing that we're supposed to do, we're supposed to explain the significance of chemical digestion of food. So one would appreciate that in the mouth, as mechanical digestion takes place, chemical digestion is also taking place, where salivary amylase is acting on starch. And of course, we understand here that it is breaking down that mechanically processed food to release the chemicals. So chemical digestion increases the availability of food nutrient to the body. It breaks down macronutrients into smaller usable form for the body. We have to have an appreciation of that as to the importance of chemical digestion. That would have covered 25 marks, which is usually the amount of marks allotted for question number 1 on your biology paper 2 exam paper. You want to ensure that you have that all covered. Here we are looking at question number 2 2AI. It has many sections, and in the box provided below we're supposed to draw a food web consisting of the following organisms. So we know the algae here, and the small fish, and the tadpole, and the water snake, and the egret. Now in doing this, there are some things that we want to do. We want to start identifying here the trophic level. So we know that algae is going to be 1, and we first trophic level, and when we are writing the trophic level, just let me show you that representation here. When we're writing the trophic level for the organism, so we'll probably represent this with a 1 and a little 0 at the top, like a 1 degree. So we want to label which one will come first, which is in the first trophic level, which is in the second trophic level, which is in the third trophic level, that type of a thing. You want to put that labeling beside these before you start constructing that food web. Now, having done that labeling there, which I was not able to do just now, you want to come on down here and start the construct. So what we know is that the algae, these are photosynthetic organism, and they are going to form the base of our food web. Then we are going to have the tadpole, which is of course a primary consumer, and this primary consumer will be feeding on the producer. Just let me show you the entire question here. Will be feeding on the producer. So you're seeing here algae coming from here being the producer, tadpole coming here being primary consumer, small fish is also a primary consumer, and then we have the water snake, which is of course getting some tadpole and some fish in, snacking, that is going to be a secondary consumer, and we have the egret pretty much doing the same. The apex predator here is going to be the eagle, and that would nicely top out this question, making it 3 marks easily we obtained just now. Now we want to know what is the role of the organism in at the start of the food web. Now this is very interesting here. When we talk about the role of the organism, we are pretty much looking at how it feeds. All right, so how does it produce or get food? So here the role of the organism at the start of a food web is going to be the producer. So it's making its own food, and it's making the food for other organism and itself, whether it be in the food chain or the food web. So it is of course making food that is of course going to be passed on to other organisms in the food web. So particularly this organism, the algae here, is going to be a producer. 1 mark for that, and we move on down to the other question. Name the organism which perform each of the following function in the food web. A draw in AI, you'll see that. So let us look at what is happening here. For the first thing, we want to look at an organism that is both a predator and a prey, or a prey and a predator. Now let us look at how is that represented in the food web. Now here, to be a predator and a prey, it must have 2 arrows. So it must have an arrow leading to it and an arrow leading away. So this organism, the snake here, is a predator to the tadpole, while it is a prey to the egret. So you're seeing here it falls within the middle of 2 arrows. Now let us be reminded that herbivory, where this organism, the tadpole, is eating the algae, this is also a predatory relationship. It's called herbivory. So between plant and animal, that is also a predatory relationship. We want to bear that in mind. We're moving on down to the other question. So we chose here water snake and egret. So we're looking at, just let me get this again, I've lost my bearings. It is both a prey and a predator. So the snake, the water snake, is both a prey and a predator, and the egret is both a prey and a predator. So we just showed you 2 examples, but I think we were supposed to only do 1, but we gave you a little extra there. A primary consumer, the tadpole is a primary consumer, and the small fish is a primary consumer. So we pretty much pulled that out there, primary consumer here, and we gave you these earlier. So let us look at the other thing that we have interest in here. We want to look at the third thing here. A secondary consumer is of course going to be the egret and of course the water snake. Remember, we were looking at that when we say primary, secondary, and then the tertiary consumer is going to be the eagle, and all that is nicely outlined here in your food web. Primary, this is going to be trophic level 1, trophic level 2, trophic level 3, trophic level 4. This is going to be a producer, primary consumer, secondary consumer, and of course tertiary consumer. As we do that, we must bear in mind the different types of nutrition. The types of nutrition are autotrophic nutrition, heterotrophic nutrition, and of course saprophytic nutrition. Now, from the food web in AI above, we're supposed to draw a food chain consisting of 3 organisms. This is really easy here because it says 3 organisms. It could have said 3 trophic level, which would have been the same thing. So we have the algae, as all food chain must begin with a producer. We have the algae, a primary consumer here, and a secondary consumer. So this nicely tops off what a food chain should be. Please be reminded that you must use the arrow to show the flow of energy. Pretty much the head of the arrow would be closest to the organism that is doing the eating. But bear in mind that an organism could be a predator and a prey. So just show that flow of energy and be reminded that the flow of energy in a food web is unidirectional. Same for a food chain. Now, what is the role of the organism to which the arrow points in a food chain? This is very easy. It receives energy from, or it feeds on the trophic level before. So the arrow that the organism that the arrow points on, it is actually feeding on the organism that is before. So it is receiving that energy to, of course, move it on to the other organism in that food web or chain. Now, explain why energy flow in one direction in a food chain. Earlier we said that it is unidirectional. Now, the producer are eaten by the primary consumer and primary consumer by secondary consumer, now causing the depletion of energy and the unidirectional flow. So it is flowing in one direction as the energy is being consumed as we move from left to right, depleted, and then it really doesn't have any organism. You really don't have a producer eating. The producer is not eating the primary consumer. The primary consumer is not eating the secondary consumer. So you find that the energy is not flowing in the opposite direction. It is flowing in the order in which the organisms are feeding. All right, so the energy reduces as it goes one way, and feeding is only done in one direction. Now, list 3 features of an environment which support living organism. Now, if we're going to be looking at 3 features of an environment, here we are called upon to pull our knowledge on biotic and abiotic components of the environment. Now, the biotic component speaks to the living component of the environment, but the abiotic does not speak to the non-living. The abiotic speaks to the physical factors of the environment. These are some factors that would be tested in geography. So here we're looking at light. That's an abiotic factor. pH, water, humidity. These are, of course, abiotic factors that we could look at. But we could look at disease, we could look at predation, we could look at competition. All of these are some biotic factors that could go nicely with these in the environment. So we have given you a couple, but please be reminded when you're answering this question, you're looking at both the biotic and abiotic components of an environment, because they would dictate nicely how an organism will survive. Now we're looking at question 3, which looks at the irritability system. One would say. So define each of the following terms. Stimulus. A stimulus is pretty much a change in the environment. A bang, you're hearing something, you're seeing something, you're feeling something, you're tasting something. All those are pretty much changes in the environment. Now the receptor is a part of the organism that detects the stimulus. So it's detecting that change. What does that mean in the environment? The receptor is picking up that something is not normal, something is off here. It's getting really hot. It was cool just now, or it was cool now and it's getting hot. So pretty much a receptor is picking up that information. Now the effector refers to the part of the environment. It refers to the part of the environment that respond to the stimulus. As the part of the environment. All right, so it's a part, the effector is a part of the individual that respond to the environment. Part of the organism that respond to the stimulus, not environment here. One might say environment depending on what you're doing, but here we're looking at individual. So it refers to the part of the individual that pretty much respond to what is happening in the environment. So it's a part of the organism that respond to the stimulus. We're looking at the eye here in figure 1 is a diagram of a section through the eye, and we're supposed to label the parts of the eye. This is like textbook. You could hop right into your textbook to get this. We're supposed to label the pupil, and it's pretty much this passageway into the eye. we're supposed to label the ciliary muscle, and here we have the ciliary muscle, and we're supposed to also label the retina. Here we have the retina here, and we're having this here at the back of the eye called the optic nerves. Pretty much are the points that we should have labeled. Now, one thing about labeling, you need to pay attention to the convention in labeling. You are supposed to have lines that are parallel to the top of the page, and you should use consistent font. So here it shouldn't be capital C or capital P. It's either uppercase only or lowercase only. So pay attention to the convention of labeling or drawing, as it were, for diagram. You don't want to miss that out. Now, why is X called a blind spot? Now, pretty much that's the area where there is no light sensitive cell, X right here, and it's pretty much a point at which the optic nerve is leaving the eye. So no light sensitive cell is in this area. It is a point where the optic nerve leaves the eye, and pretty much that's the answer for that. There's no further explanation for that. We're looking at this. A boy looks up from his book to see a helicopter flying in the distance. Okay, great. Account for the changes that would occur in the lens of both of his eyes to enable him to see the helicopter. Here we are called upon to look at the ciliary muscle and the suspensory ligament. So the ciliary muscle will relax while the suspensory ligament will pull tight on the ciliary muscle. Thus, the lens is pulled tight by, of course, is pulled tight by the suspensory ligament. Now, this, of course, will allow the lens to bend light rays onto the retina. So this is what he's going to do in adjusting from seeing near object to seeing distant object. You can use a diagram to enhance this question if you so desire, as you are pretty much describing or accounting for what happens here. The other thing that we need to do here for figure 2 is to show two labeled A and B under two different light conditions. We need to read all the instruction. Eyes under different light conditions. So we have an eye at A, we have a eye at B, and look at the pupil here versus the iris. Look at the size there. Which of the eye in 2, A or B, shows the appearance of the eye exposed to dim light? All right, so this is the eye here in dim light. It will actually open the pupil there, allow more light to get in. Why is it important for the eye to pretty much respond when exposed to very bright light? All right, you have to protect the eye from damage that can be caused from this light. So this heavy light entering the eye will, of course, damage the light sensitive cells. You really don't want that to happen, so the eye need to respond in order to protect itself. We're on to section B of a paper, and we're supposed to answer all questions in this section. Write your answers in the space provided in this booklet. In the box below, draw a label diagram of a female reproductive system, and using an arrow, trace the path that spermatozoon would have to take to fertilize an egg. So here we know that the egg is actually fertilized in the fallopian tube before moving down to the uterus. So here we would have done some drawing, and I have used Microsoft Word to do this drawing. So if it is not like the one in your textbook, forgive me, but I do hope that the concept is taken. So here we would have, of course, the title, and usually your title is underlined. So it is nicely underlined there, and of course written in uppercase. Again, here the lines are supposed to be parallel to the top of the page. Some of my lines are not, but I do hope that the concept is learned as I teach. So here we have the oviduct. Look at the labeling convention. We are making sure that all the letters are pretty much the same case. Everything is lowercase. So labeling, and we are labeling on one side, and we wanted them to be in a straight line, but I don't think I am maneuvering Word very well in doing this, but I think you understand clearly what is happening from my drawing. And the arrow here traces the part of spermatozoa, saying that right about here in the fallopian tube we would have fertilization taking place. Make sure that you can do that in your experiment or in your exams. Now we move on down to the other question. A young couple wanted to choose between two contraceptive methods, alright, barrier and surgical. Discuss why the male may prefer the barrier method rather than the surgical method of contraception. Your answer should include 2 advantages of a barrier method and 3 disadvantages of a surgical method. He would prefer the barrier method as it is very reliable. It is easy to use and of course readily available. All these would suit his need in a contraceptive. While the surgical method presents the challenges such as being expensive, requires surgery, and is usually irreversible, these would have served as limiting factors that convince him to use the barrier method. So sticking out here is that it is expensive. It will, of course, you require surgery. It might be irreversible. And a lot of persons are afraid of knives and surgery. And you want to protect his manhood. I really don't want to feel like, hey, no, that's not the thing for man. So they'll prefer the barrier method at all times using a condom. Remember though that using a condom, or in most cases, will protect you from a lot of STI. The surgery might not protect you from STI. Always remember that the surgery doesn't really protect you from STI. It, of course, protects you from pregnancy. A biologist discovers that the flower of a species of plant have no male reproductive parts. So just one method by which these plants produce offspring and 3 advantages of this method. Now, of course, this plant is reproducing asexually, meaning that it is using a part of itself rather than using seeds. Now, the advantage of this method is that if they are adapted to the environment, all will survive when it chooses to reproduce. The process is rapid as no mating time is needed. The process is also energy efficient and maintain desired characteristic if there is, of course, desired characteristic. We're looking at a croton and you were supposed to pull a part of that stem and plant it, you'd get the exact croton, so that desirable trait would have been maintained. And we move on to question number 5. With the aid of chemical equation, explain what is meant by the term photosynthesis and discuss how a dicotyledonous leaf is adapted to capture maximum sunlight and carbon dioxide for the process. Now this is like straight textbook, and I really didn't expect the question to come as it is here. Photosynthesis is pretty much a process in which autotrophic organism converts simple inorganic substances to organic substances using light energy from the sun. I know that you were expecting this primary school definition, but stick to the definition that the syllabus is arguing. The equation is going to be 6 carbon dioxide with 6 H2O, that's water, to glucose and oxygen. Once it's written like this, it must be balanced, which means that the amount of carbon on this side must be the amount of carbon on this side. So I'll quickly balance it with you. So here we have 6 carbon. This 2 does not apply to the carbon, it only applies to the oxygen. So 6 carbon here, and we also have 6 carbon here. On this side, we have a total of 6 oxygen, and this 6 applies to this oxygen. 6 times 2, it's going to be 12 here, plus another 6 oxygen from this 6 here, it's going to be 18. Let us test this side. 6 2 is 12, plus 6, 18 oxygen. Let's look at hydrogen. 6 times 2 gives you 12, so we have 12 hydrogen on this side. If you observe this 12, the subscript here, 12 hydrogen. So you see that the experiment, it is pretty much balanced. The equation is balanced on both sides. All right, same number of each on both sides. You want to ensure that you have that if you have to write the equation and not the word equation. Now, the adaptation of a leaf of photosynthesis, this is textbook, and a lot of things can be said. Now, the leaves are broad and flat, and they have this flat lamina that catches light readily, They are thin, hence allow light to pass through very quickly. They lie at 90 degrees to catch adequate sunlight. They have stomatas for gaseous exchange. The leaf contain chloroplasts to, of course, trap the sunlight. And we have xylem vessel that is transporting water and mineral, and phloem vessel that transport pretty much a food nutrient to various parts. We can't forget, however, that the leaves have a waxy cuticle that reduce the rate of water loss from the leaf. So all these are adaptation of a leaf to carry out photosynthesis. I would have never expected this question to be asked in the way that it is asked. It is valid 11 marks. That is a big thing for student. More so if you were studying all over the place and you did not zero in on so much. You probably just study about 3 or 4 for photosynthesis. But this is a cue for us to be prepared for the exam. Now outline how decomposers assist in cycling carbon dioxide to make it available to green plant. Now decomposers, they really cycle this carbon dioxide as they respire during the process of breaking down organism, breaking down organic compound. So nutrients pass through the ecosystem from autotrophic to heterotrophic and finally to saprophytic organism, which decomposers are a part of the saprophytic group. As the energy move, organisms acquire organic compound which is stored in their bodies. When they die, excrete or ingest, decomposers, fungi and bacteria, these are your decomposer, they feed on the remain, producing carbon dioxide through the process of respiration, which enters the air and is used by plant for the process of photosynthesis. So they are breaking down everything as they aid in this decomposition, releasing that trapped nutrient and energy that is pretty much in this food. It's released into the soil, taken up by plants, and of course the carbon dioxide pretty much enters the air to be a part of the carbon cycle, continuing the cycling of carbon. And we move on to question number 6, which is the final question, a pretty interesting question. State 2 causes of anemia and describe 2 signs and symptoms of this disease. Now the causes of anemia, there are so many, but the first one will pop on anyone's lip is iron deficiency. If you have cancer, you could have anemia. If you have bone marrow failure, that could also cause anemia. Now signs and symptoms include you'd feel weak, you'd feel tired, you might have cool hands and feet, you'll become dizzy, that's a popular one, and you'll have headache. So feeling weak, dizzy and headache are some of the common signs of anemia. So we want to move on down to sickle cell. Now sickle cell anemia is caused by the presence of a recessive allele. Alright, persons who are heterozygous and carry the sickle cell allele are more resistant to a type of severe malaria than persons who are homozygous for the normal hemoglobin allele. Now state what is meant by each of the following terms. So homozygous here means having identical allele for a certain trait. Heterozygous having different allele for a certain trait. Now let's move on and look at the other part of this question. Now here with the aid of a genetic diagram, show how a man and a woman who do not have sickle cell anemia can have a child who has the disease. Now first, there are some things that we need to do to solve this, and I'm going to show you everything on the screen that you need to do. Alright, in the box is everything that we need to do. First, we have to define the alleles. So we are going to define, let N be the normal allele and S be the recessive allele for sickle cell anemia, for the sickle cell disease. Now one of the things we need to know now is the rule for acquiring recessiveness. And it states that if you are going to have the disease, you must acquire the trait in its homozygous recessive form, meaning that 2 of the same allele. But the first thing we need to do here is to, like I said, define the alleles, and then we are going to write out the parental phenotype. So both parents were carriers as they were heterozygous for the trait. The next thing we're going to do is to write out the genotype. Now you are seeing HB here, and HB just speaks to the hemoglobin or the locus, the point where we are finding this recessive condition. So we have the genotype being written as NS, that's the allele that we're using there. So it's HBNS, and the NS is pretty much a superscript. The next thing that we need to do is to write the gametes from the genotype. Now the gamete is just to separate the two alleles, as you're seeing here, N and S separated on both sides, and then we are going to have segregation. Now it is at this point you might do those cross things, as a student call it, or you might use the Punnett square. Here we are using the Punnett square, and it doesn't really matter whether it's a male or female, we're just looking at recessiveness. So what we did, we placed one set of the N and the S here, and another set of the N and the S here. And please be reminded, you can go to my channel, Csec Biology TCP, and view my playlist on all your genetic crosses. Alright, please also be reminded to like, share, and subscribe to Csec Biology TCP. Be reminded, I am Mr. Wilson, and we have over 300 videos for your perusal. So let's look at what we did. So looking at the screen, N and N intercept here, and what we did, we wrote both the genotype and the phenotype in. So the NN represents the genotype. This is going to be the possibility of one of the offspring. We are saying this is a normal person. This person doesn't suffer from sickle cell. N and S intercept here. This is the genotype. NS is the genotype. So this person is a carrier, and that's the person's phenotype. Alright, it's never usually written in the cell like this, but we did this for you to follow pretty much a lesson. N and S pretty much intercepts here, so what you find is that this is also a carrier. And here we have the homozygous F1. So here it's a sickle cell child, as the trait is inherited in its homozygous recessiveness, SS. So this is one person here with sickle cell. Now it's important to look at the fact that in writing the phenotypic ratio, we have 1 normal to 2 carrier to 1 sickle individual. Now another thing that we need to look at here is that the possibility of 25% of the offspring will be normal, 25% of the offspring will have sickle cell, and then we are going to see a 50% possibility of us having a carrier from this interaction between these two carrier parents. Now we're supposed to explain why the sickle cell allele is more common in the population in countries where the survivor of malaria is present. This is very, very interesting here. If we look back at this Punnett square, we're going to see some of the answer there. Now the carrier of a trait, and this is the heterozygous form, are normally naturally selected as they show resistance to malaria. Once you have the trait, you are resistant to malaria. This allows them to pass on the trait to offsprings, and that's a 50% pass on for you to be another carrier. While other persons might have died from malaria, removing it from the gene pool, when two heterozygous parents parent and offspring, it gives a 25% chance of the disease being passed on and a 50% chance of the trait being passed on, which allows sickle cell to be common in areas with malaria, as the trait is persistent because it seems to be naturally selected, and persons with the trait are usually resistant to malaria. Hence malaria continue to live or to persist or to pass on from generation to generation. This takes us to the end of this 2016 biology paper 2. Please be reminded to like, share, and subscribe to Csec Biology TCP. Please enjoy our many past papers for biology and human and social biology. We also have labs, animation, notes on the channel, enough videos for you to pass your exams. Thanks much for joining. All the best in your exams.